Well, thank you. I am a biochemist working on AADC protein. AADC protein is an enzyme that catalyzes the conversion of L-dopa and L5-hydroxytryptophan into dopamine and serotonin. It is synthesized by a gene located on chromosome 7, on our chromosome 7, and each time there is a mutation in this gene, you will have a protein that shows a defect. A defect means that an amino acid is converted to another one, or you can have a deletion, you can have a stop codon, so at the end you have an altered protein. And this altered protein determines the presence of AADC deficiency. This means that these patients have a sum of symptoms that could be related to the misfunctioning of this enzyme, because the problem with the enzyme is that an alteration in structure determines an altered function. And which is my study, my research is devoted to understand the molecular basis for this structural defect. I mean that I go to study why this alteration, why this mutation determined an impact in catalysis, that is in dopamine and serotonin production. And for example, I can find that a defect determines a problem in folding. And this is useful because we can think about chaperone therapies for these patients. And the rare disease patients need to be addressed with personalized therapies, because even if the symptoms are similar, the defect could have a different molecular causes. We can do predictions. We can predict that if a mutation maps on a protein region can determine a defect different from another mutation mapping in another protein region. And we can also examine the protein population possessed by the patient, because an homozygous patient has a different protein form than an heterozygous one, which is much more complex because it has much more defects. So our approach, our study needs to be, of course, validated in appropriate cell models, we should talk at in strict relationships with clinicians, because we have need to compare the phenotype of the patients to the phenotype of the misfunctioning enzyme we have in hand. And I think that this integrated manner to perceive this disease could be very helpful in, foresee appropriate therapeutic intervention to individual patients.
Okay. Can you name or count benefits for the patients within this enzyme you're mentioning?
Yeah, I can see that our molecular studies could be the basis for ameliorate treatments that now are given irrespective of the genotype type to all patients, and this could help clinicians to focus on some treatments more than on others. For example, if we have a mutation that hit the DOPA binding site, it's important that in these patients there should be a supply of DOPA could be beneficial to these patients. If we have a patient that has a mutation in an important and necessary amino acids that is chemically important to perform catalysis, so L-dopa has no benefit, the addition of L-dopa. So we can work in collaboration with clinicians to find the best way to treat the patient. So I think this could be a benefit. Prediction and precision medicine therapies could be addressed by the combination of these approaches.
Great. Thank you very much.
Thank you.